Hello everybody, thank you for coming to this first talk about ART IBMCP, the art testing residence that we have set up here in the IBMCP together with the Vice-Rectorate, the collaboration and the supporting of the Vice-Rectorate of Art, Science, Technology and Society of the UPV. First of all, I want to apologize in the name of Pablo Vera, our director of the IBMCP. He has not been able to be here because he has a last minute meeting with high people in the so he wants to apologize for not being here. And then I want to introduce Salome Cuesta, the Vice-Rectora, I don't know how it's said in English, the Vice-Chancellor of the Vice-Rectorate of Art, Science, Technology and Society. And I want to thank her for being here, and I want to thank her for the collaboration and the supporting of this first artistic residence in the UPV. So, well, welcome, Salome, and I think she has something to tell us. So, when you want, cuando quieras, Salome, puedes pasar y contarnos tu...
Buenos días a todas y a todos. Gracias por haber acudido a esta primera jornada, seminario, no sé muy bien cómo lo llamáis, charla, donde vamos a tener la oportunidad de escuchar a Carlos Castellanos, que es el primer artista en residencia que tenemos en este nuevo programa. En primer lugar, a mí me gustaría contaros un poco, compartir con todas vosotras, ¿no?, cómo surge este programa. Sabéis que hace más o menos un año se produjo una elección, las elecciones a rector aquí en la universidad, y el equipo de Pepe Capilla, del cual formaba parte, bueno, pues ganamos las elecciones. El hecho de tener un vicerrectorado de Arte, Ciencia, Tecnología y Sociedad es algo que yo le propuse al rector como una novedad, puesto que hasta ahora sí que es cierto que el vicerrectorado era o de cultura o de acción social, pero no había un vicerrectorado que de un modo interdisciplinar agrupara los distintos modos de conocimiento que se producen en la UPV, ¿no? Somos la única universidad politécnica en España que tiene una facultad de Bellas Artes y también estudios de ADE, y creo que esa particularidad, esa excepción que tenemos aquí en este campus, deberíamos aprovecharla, ¿no? Y aprovecharla con iniciativas que ya sabemos que en otros ámbitos internacionales ya estaban testeadas, como son las residencias de artistas, para trabajar, para colaborar con los investigadores en la búsqueda de una nueva cultura científica que creo que, por la formación que tenemos todos aquí en el Politécnico, estamos muy abocados a desarrollar, pero que se produce en muy poca medida. De modo que esta residencia, este programa de residencias de artistas en laboratorios científicos, lo hemos planteado como una estrategia que el rector apoya, que busca socios colaboradores, como es vuestro instituto, y que de verdad que os doy las gracias a todas vosotras y al director también, a Pablo, aunque no haya venido. Y pensamos que es una estrategia para poner en valor todo el trabajo de investigación, de transferencia que estáis desarrollando, haciendo como un salto, pasándolo al plano cultural, y de hecho los resultados de la residencia podrán exponerse, podréis contemplarlos y disfrutar de ellos en el centro del Carmen. Creo que ya habéis estado interaccionando con Carlos, algunos de vosotros. El proyecto que surja, yo creo que poder mostrarlo en el centro de la ciudad y aproximar toda la investigación y el trabajo que hacéis a la ciudadanía es uno de los objetivos de este vicerrectorado, y vamos a apoyar porque todo esto ocurra. En ese sentido tenéis también, aprovecho para hacer aquí una cuña de publicidad, la nueva exposición que podéis ver en el Ágora, en el edificio nuevo, se llama N-1, y es una exposición que se titula Futuritat, la vida dels materials, para que podáis comprobar también como todo ese trabajo de investigación que hacéis en los laboratorios, si se saca de contexto y se pone bajo una nueva luz, cobra un nuevo sentido. Entonces, ayer la inauguramos, hoy por la tarde, está aquí la directora de área de acción cultural, María José Martínez Pison, que ha sido un poco la responsable de coordinar todas estas acciones. Desde el Vice-Rectorado queremos apoyarlos en todas las acciones que penséis que pueden contribuir a sacar todo ese conocimiento a la ciudadanía. De modo que tenéis las puertas abiertas para plantear proyectos con nuevos enfoques disruptivos, experimentales, y que trabajemos todos en acercar la ciencia a la sociedad. Muchísimas gracias a Javier, que ha sido nuestro interlocutor, y sobre todo a Carlos, que es nuestro primer artista en residencia en este programa, y queremos trabajar a fondo con él para que salga un proyecto inmejorable. Muchísimas gracias a todos por venir. Gracias.
I don't mess up with this. It's just a few words. Now I want just a few words to put to give context for the artist residents in a scientific center, okay? Because I think, well, I know that some of you, or maybe many of you, are thinking, what the fuck is this? An artist in a scientific center? What is this? Why? Why? Because artists are crazy people. That they do crazy and weird things, and scientists are logical people that do things that have some utility for the society. So why an artist in residence in a scientific research center? Why? And the short answer is just for fun. Okay, just for fun, because as everything, almost everything we do in life is just for fun. Okay, this is true. Well, not really. No? Neuroscientists tell us that we humans make decisions first based in a rapid and fast emotional response, and only after that the critical thinking, the logical thinking comes to the rescue in order to give us a justification of our previous emotional decisions. And this critical thinking makes us to believe that we are doing this decision because we are thinking logically and with rational thinking. And so we believe that really we have the things for not just for fun, okay? And then I think there are two main. Salome has told us, more or less what is the reason of having a collaboration of artists and scientists, but I think mainly the benefits are twofold. The first one is to gain visibility for science, for research centers, for scientists, okay? Because of course we all know that it's very difficult to get a time slot, a lot of time in the mass media, no? In a television, a national television, for example, okay? But for science news, it's even harder. Of course, other than science news related to the heat of the moment, like pandemics and volcano eruptions and something like that, right? Okay? But scientists are more easily, they reach people more easily. So they can communicate a lot of things in different ways that we scientists we don't know to do that, or at least most of us we don't know. Okay, but artists are trying to communicate, to give different representations of the things. Okay, and not only that, the second reason is that art can the usual role of art is give different approaches to the reality, give different concepts to provide different perspective of the world, of complex issues. Okay, and well, I don't want to say a lot because some people in say that before better than me, so you can say that they say that artists are more creative and represent the data in transformative ways. Okay, and art is important to communicate abstract and higher order ideas. Okay, and we live in a world increasingly complex. Okay, so we need increasingly more divergent approaches to the things, or more divergent and new and different approaches and different concepts. Okay, so there is a lot of interest in the incorporation of artists to almost any field in the world. Okay, for example, one typical example is the shift from STEM to STEM. Okay, STEM is science, technology, engineering, and mathematics, and this is always a lot of institutions trying to the people get involved in these fields. Okay, and then recently they have added the A for art in these capabilities that we have to develop to go to the world. Okay. Also, there are a lot of they are setting up a lot of centers that are specifically dedicated to the work with art and technology and science. For example, the one from the MIT, the Massachusetts Institute of Technology. Also, there are specific journals. Okay, this is a wide community of people working in this interaction on art and science. And they have, for example, this journal is the leading journal on art and science collaboration. Also, there is a lot of festivals, international festivals, where people working in this show their work. For example, the Ars Electronica, that is more or less the leading festival on these issues. Even in institutions, in state institutions, there are a strong interest in promoting these things. Okay, so there is this institution in the United States to promote the incorporation of art in the research universities, and in Europe we have the STARS program, Science, Technology, and Arts, that is funded by the EU, the European Union, in order to fund and to promote the incorporation of artists in research centers and other things of collaborations between arts and scientists. Okay, and then there is a lot of companies and research centers having, as we are trying now, artistic residence. For example, one typical example and very known example is the CERN, the Particle Physics Institute European, that have a lot of years, high register of years, having not one, several artists all the year around, messing up with the scientists, with the labs, and doing artworks by in his work. But not only the CERN, Max Planck has also the Fermi Lab, a lot of universities, the European Space Agency, and a lot of them. Okay, and not only research centers, also companies, also private companies are doing that. Here in Spain, there is not a lot of initiatives like that. So we have, again, thanks to Salome and the Vice-Rectorate of Science, Technology, and Society. One known example is this one from the CNIO, the Cancer Research Center in Madrid. Okay, that has 4 or 5 years having artistic residence, and even in the last one, they even get a stand in ARCO, the Festival of Contemporary Art. The first time that a research center has the wide visibility provided by a festival like ARCO, okay? Apart from this center, I only know these two ones that are also excellent centers. That are the two centers of Barcelona. The Center for General Regression and the Institute for Research Medicine. And so we were very happy to participate in this international effort to provide new ways of looking at the problems and new ways to the art and science collaboration. So that's all I want to know. Now we are going to introduce Carlos Castellanos, that is now our artist. Is from the United States. He's now in the Institute of Rochester, and he has a long track of working with science and technology, and he will tell us about that. Just for the end, if some of you are interested in participate, collaborate with Carlos, feel free to contact us or contact him directly to talk and to propose any proposal. But remember, it has to be just for fun, okay? So thank you very much, and Carlos, you are welcome.
Okay, thank you, gracias. Entonces, yo voy a hacer la charla en inglés, pero si después en el Q&A me quieren preguntar, hablar en castellano. Y gracias a Javier y Salomé y María José por esta oportunidad. Okay, so, yes, hi, I'm Carlos. I know some of you already, and I hope to get to know the rest of you throughout my time here. So I'm going to talk about intersections of living and machine agencies, which is, could be almost anything, right? So I'm interested in kind of those interfaces of humans and non-humans. And again, non-humans could be almost anything, right? So I'll be, sort of discussing my own work and my own sort of kind of angle on that. Really quick, just a little bit of background. So my original background was actually more in music. And that's sort of how I got into technology. Right? And so I have, but my undergrad and my PhD and my MFA, they're all in kind of new media art. And I'm, as Javier said, I'm in Rochester Institute of Technology in the US. For those of you who don't know, Rochester is like in upstate New York. It's kind of near Canada, a couple hours drive from like Toronto. And yeah, so I kind of had like a previous life, like I said, kind of in sound and music. I was an auto engineer, and then I moved to San Francisco, and I was a web developer. So I've taken a kind of strange route to where I am today. I'm not sure actually how this happened, but it's for fun, right? So, okay. So I approach my work as kind of a sort of science research, or kind of an autonomous zone of research, I guess you could call it. So my sort of interests lie kind of at these intersections, right? And I'm not, of course, the only artist working in these areas, but I have this kind of, in my view, kind of a a weird sort of combination of these three things. These are the sort of the areas that I'm interested in. And I started kind of, like I said, in sound and music, and it was more like, I got into the technology through that. So I was kind of, I'm just going to say like a programmer or a developer. So all the other stuff kind of came later. And, you know, again, I'm interested in these things as a cultural practice. And what I'm interested in is these kind of notions of complexity and adaptation and emergence and, you know, like collective intelligence and, you know, all these kinds of things. And I think what sort of brings all these elements together, I think, is that they all kind of thematize these things. They thematize emergence and complexity and the kind of, you know, human, non-human intersections, and, you know, there's, you know, the different cultures and knowledge systems and all that are involved. So again, you know, this is my sort of can encapsulate maybe in a couple of images what I'm talking about. These are, of course, this was like, I didn't do it, but these are like, this is a little bit photoshopped, as you might be able to tell, but these are bacteria, right? These are Physarum polycephalum. Some of you probably know what I'm talking about here. These are plant roots that are being used as logic gates, right? So this is like the world I'm interested in. So these, the slimologists, you know, they've used them to like model like transportation networks and subways, and they're much better than like anything like an engineer can do, apparently. And I've worked with these pattern-forming bacteria. I'll talk about those in a minute. And there is kind of an emerging field of plant-inspired robotics. We can talk about that in the Q&A if you want. I don't follow it too much, but this is kind of the complexity, you know, how Javier was talking about, these kind of intersections. And yeah, so and this is, to me, this is already kind of the beginning of what I'll talk about in a minute, a sort of art science, or a new kind of science or art science. It's more of a complex adaptive systems view of life and intelligence, right? Again, more of a focus on adaptive relationships with the environment, an informational, cognitive characterization of the living. For me, all of this, by the way, comes from cybernetics. I'll talk about that in a minute as well. And a lot of my work is about these kind of embodied flows of information, right? Embodied meaning it's like there's a sensuous presence to it, right? It's not, we think of information as disembodied, but I think of it as very embodied. And I'm probably preaching to the choir a little bit here, but that's again, just context. So as I said in the abstract, what I want to sort of, I'm not going to get into too much of a like theoretical discussion. I'm mostly, I think, going to talk about my work, but I do think there is kind of an emerging aesthetic paradigm here that's rooted in this kind of complexity, in the way that non-human organisms, typically quote unquote primitive, like bacteria, plants, etc., how these organisms and intelligent machines may evolve their behaviors in response to one another. Like there's a there, I think, right? So again, it's just kind of alternative models of how we think about complexity and intelligence and all these things. There's a lot more I can say, but I'll leave it at that. And just, again, for a little more historical context, so I don't know if anybody recognizes this image, but this was from MIT Media Lab in 1970. So this is a project where there was a computer scanning these blocks. I don't know if you can see the gerbils here, pebbles. So that's sort of their environment, and so the idea was that this was a computer-controlled environment that would manipulate the blocks to where they thought the gerbils might want them. But of course, the gerbils then reacted, and again, we'll get into like the whole thing about it, but basically the designers didn't really anticipate what the gerbils were going to do, so the reactions were unexpected, and it was sort of kind of like it's sort of like the gerbils were outsmarting the computer, and everything was kind of just like a mess, like a disarray. So I'm not as we'll see with all the other work, not so much interested in the results, but that process, right? This was in like 1969, 1970, and I show this just to give you sort of a sense that even from this early days of what you might call new media art, there was already kind of the non-human world was already kind of involved in some way as a source of complexity and variety, right? So the project is called SEEK, and it was 1969. Okay, so let me talk about my own work. So this first project actually doesn't involve non-humans, not specifically. So this is a project that I did with a neuroscientist friend of mine from UBC, University of Columbia. It's called Biophysicist, and it's based on the work of a cyberneticist called Gordon Pask. So this is, again, kind of the same theme. This is really my first project, like truly investigating this kind of stuff. So this is kind of an electrochemical computer, sort of like that. We're growing wires here, essentially. But it's based on the work of Gordon Pask, who was creating, this was like in the 50s, who was creating, basically, this is before digital computers, so that he was creating kind of, they call them fabrics, or basically substrates for control systems, or what we might call computers. And this was kind of like a self-organizing, self-repairing system that actually was able to learn aspects of the environment. So it's, we don't live in this world, right, where we have like electrochemical computers or computers made from slime mold, at least not yet, right? But that was a possibility, like people were looking at this back then, right? So what is it? So we have a shallow bath of stannous chloride and alcohol, ethanol, and so it looks, basically clear. But when you put in an electrical circuit, it grows these wires, right? These kind of fractal metallic like patterns. And it turns out, so we did a kind of a definitely pseudo-scientific experiment, kind of an associative learning experiment, where we said, can we associate something happening in the environment, like movement in space, can we associate it with some growth, which is what Pask did. He was able to steer is the better word, train it not like you train machine learning, but more like adaptive steering, to actually be sensitive to certain elements of the environment, like sound and magnetic fields. So it would respond to sound and magnetic fields in a certain frequency, like 50 hertz. Okay, so we kind of recreated his experiments, right? So these are the electrodes. So we have this kind of fluid, sort of network with, it's just anode and cathode, and we put in the electrical current. And then we have relays to turn on or off some of these electrodes based on, again, information from the environment. So if there was movement so we track movement with like a camera, like a webcam. So if there was movement, let's say this area of the space, this electrode would get, this relay would trigger, and it would close the circuit between like an anode and a cathode, and then you would get growth, right? And some interesting things happen, right? We're putting in sound like in different frequency ranges, so like splitting the sound into frequency bands and putting those into electrodes. And we were saying, yeah, can it associate maybe movement and sound, movement in a certain space with sound in a certain frequency range? And we don't have any like results, but other than this was a very like activity dependent thing. So these things would also dissipate if there wasn't activity in the space, right? So it was, I'll just say it was kind of interesting. And there's time I'll try to play all the videos of my project, but I'm not sure if I'll be able to. But let me play a video for this one. Which is here, yeah. And I don't know. Oh, there it goes. Yeah, it's cutting out like it was before. Yeah, lo mismo It doesn't matter, it's just me speaking, basically saying the same thing I just said in kind of fancier language, but you can see, so it's a very, it looks like a very DIY setup, and it kind of was in the early versions of it. So it's also kind of like a repatchable thing, so we could actually change what electrodes are connected to what, just literally by patching, kind of like a audio patch base. So you can see some time-lapse growth in a couple of the electrodes. Yeah, okay, so I'll leave it there. And yes, so as I said, this is based on the work of a cyberneticist called Gordon Pask. So here was kind of a group of, I know when we think of cybernetics, at least from the United States, it's very much tied to like the military-industrial complex. And that is true to a certain extent, probably more like operations research. But there was this group, primarily British cyberneticists, who were kind of doing this weird stuff. What Pask was interested in is, I mean, they were kind of developing a different way of doing science, really, that tried to actually account for the observer. In other words, it was trying to account for subjectivity, which, what is that? You're not supposed to do that in science. But he had this notion of what's called a participant observer, which is different from a cybernetic, the participant observer is different from the scientific observer in that he or she or they will actually want to interact with the thing that they're studying. Typically, you're not supposed to do that, right? Because you're supposed to be objective. So the idea, as he says here, is a participant observer decides upon a move which will modify the assemblage, and in general, will favor its interaction with it. And that's what he was doing with these electrochemical systems. That's sort of what we were sort of featuring, right? And Andrew Pickering, who did kind of a history of these guys, he says this is sort of the possibility of a basis for a new kind of art science. So I'm going to come back to this. But let me talk about a few other projects. I'm also interested in like environmental health and bioenergy technologies. So this is a project I did in China, in Wuhan, in 2016, back when nobody heard of Wuhan. I mean, I had heard of it, but I didn't really know hardly anything about it. So this was like a wonderful experience for me. So basically, it's a floating wetland with a bunch of sensors on it that monitors the water quality. And we deployed this one. We deployed in the lake near the art center, the art village where we were staying. So it was a residency, kind of like here. So I was doing work, I was doing workshops, and eventually this was the exhibit. Technically was in Hong Kong, but I also did this kind of, I want to say, guerrilla exhibit. Didn't really ask permission. We just deployed it in the lake. So we worked with some of the people there and some of the other residents. So I'll talk about it a little bit more in a second, just to give you a little context. So here's Wuhan, Shanghai, about 800-900 kilometers from Shanghai to Wuhan. And Wuhan is on the Yangtze, and there's a lot of lakes there. And they've had, it's better now, but they've had a lot of issues with water quality, and with basically just losing their lakes. They've lost, because of development, probably about 30 to 40% of the lakes there. But even still, so this is Wuhan. So even still, you can pretty much walk anywhere, and you'll see a lake. So that image with the lake, that was near the art village, and there was a bunch of apartments. So the idea is that, what if you could just walk? You're walking home, and instantly you can just see a set of lights that'll tell you kind of the status of your lake, even if it's just, like I said, the water quality. When I say water quality, we were measuring pH, dissolved oxygen, temperature. So yeah, so it's a combination of environmental sensing, mobile technology. So these things were connected to the internet, and they were solar powered. And long term, I was maybe interested in a kind of interspecies communication. Never really did it. In other words, there's fish in it, can we talk to them somehow? But really, it was about fostering sort of community agency in addressing environmental, in this case, water quality issues, right? And kind of exploring alternative models of citizen participation and engagement with environmental science. And sort of the vision would be maybe you could have like a network of these across the city. So just real quick on the kind of implementation. So it was just these constructed wetlands that were sort of floating, and the sensors. And it was all solar powered, like I said, and there was a like a little 2G like cell phone module that would send the data to the cloud. So we were kind of visualizing in real time. I wanted to make a WeChat bot so people could get on WeChat, but didn't have time. So it was just the internet. And all the data was recorded. This is sort of a screenshot. If you don't know what this is JSON. JSON data. So it was shared. Anybody could have access to it. And like I said, we did an exhibit in Wuhan where, in Hong Kong, where we took native plants and just brought them into the gallery and basically did the same thing in the gallery. But really the fun part was Wuhan. Okay, so that's that. I'm also, as I said, interested in bioenergy technologies. I've worked a lot with microbial fuel cells. This is a piece where I'm basically sonifying bacterial voltages in microbial fuel cells. And basically, I would say it's an inquiry into bioenergy technologies as a kind of, as a artifacts of cultural exploration, right? The basic idea here is that we're giving the bacteria a voice, right? So we can hear them in some way. And more broadly, I would say, if we can actually, well, I'll show you in a second, but it uses machine learning to kind of map the life stages of these bacteria as understood through their voltages. And the idea is if you can actually get some kind of understanding of those bioelectrical patterns, you can, this could be almost like a tool, a sonification tool, or even a compositional tool. So microbial fuel cells, they're just batteries, right? So anaerobic bacteria, some anaerobic bacteria, actually release electrons when they metabolize food. So you basically, this is what's called a two-chamber design. You have an anaerobic side, which is the actual, so you just get like mud, benthic mud, from the bottom of lakes or even compost, things like that. And on the other side, you will put an aerobic chamber. So this is anaerobic, this is the aerobic with just water, maybe with a little salt. And then, if you have a lot of sort of good bacteria, you'll get an electron flow. Obviously, it's very low voltage, in the millivolt range here, but you can boost them if you want. And you have this semi-permeable, what's called a proton exchange membrane to sort of positive hydrogen ions. You can also do a single chamber design. You can buy these like on the maker store, right? These kind of things. They're really easy to build, or like I said, you just buy them. So this is the same idea, but it's a single chamber design. So the cathode, this is what's called an air cathode. It's just right sort of on top of the water. So the project involved actually connecting these, this is sort of a different version of it, to modular synthesizer. So I'm a synth, as I said, I have a background in sound and music, so I'm like a synth nerd and a drummer. So basically, we won't get too much in the weeds here, but basically I connected them to the synthesizer. They're sort of like kind of like a control voltages. Using Eurorack synthesizers and a very simple machine learning model to map what I call the life stage of the MFCs to sound. And I had, in most of the installations, I had 4 channels, 4 MFCs. So it was 1 sound per channel. So basically I created kind of like a smart mixer to mix different sounds. So this is what I mean by the sort of a life stage. When you create an MFC, it'll last. I've found a lot of variety, but this is a sort of 45 to 50 day lifespan. I've never really had to live that long, but what I did is basically I took this mapping and I remapped it to think of this as like a mixer with 4 channels. So it's just a simple regression curve. super simple, like no deep learning, nothing, just a simple like neural network. I guess shallow learning. And this is sort of kind of the system. So the microbial fuel cell, we're maybe amplifying it. I don't remember if I actually did amplify it or not. And it can be connected straight to it's a software called Max that I use for like interactive installations. Or I can also connect it to the Arduino itself, an Arduino that would actually measure the voltage. So either an Arduino or directly to the six. And these are all kind of, maybe some of you know what these things are, like pitch shifters, envelope followers, all these kinds of things. And the piece works on 2 temporal scales, right? So you do sometimes get like the transients in the voltage, so this sort of mapping those, and then this longitudinal, the 48-hour thing that I talked about before. Go ahead and play the video for this, but there's no sound. I don't know why that's happening. But this, as you can see where how it was set up. So there's 4 MFCs, 1, 2, 3, 4, and then 4 speakers. And this is just, it's mud. I forget. This was at University of Washington. We just got like compost from their recycling, from their compost. Okay, so I have worked a little bit with plants before coming here, but in a pretty different way. So MFC. You can also make MFCs from plants. So that's basically what I did for this project a couple years ago. So so it's called Plant Connect, and we're sort of asking how can we use new media, computational technologies to create systems that sort of again, interface with non-humans and create kind of novel, I want to say interspecies experiences. So this project, we're basically measuring the photosynthetic and bioelectrical activity of plants. There's plants here, there's rice plants, Asian rice or Rhiza sativa, and sort of, again, translating them to sound and light patterns using PMFCs, plant MFCs. So we're using biosignals. We're measuring CO2. That's a way of measuring photosynthesis, right? Because they absorb CO2 when they're photosynthesizing. Computer vision to track the light patterns. A little bit of machine learning to cluster the different light patterns. And then we're sending them to an 8-channel surround sound system. Okay, so we got these plant-based microbial fuel cells. So how does that work? Well, they photosynthesize, they release the sucrose, and that sort of feeds the bacteria, essentially, right? And we're still getting the voltages from the same bacteria, sort of at the bottom of the soil, because they're anaerobic, so you don't want to give them oxygen. Okay, so we're measuring them, and then with a microcontroller, and then again, I won't get too deep in the weeds for the lack of time, but I'm looking at sort of like the minimum and doing some analysis, so the minimum and maximum ranges, and that will determine what lights go on. When I say lights, it's grow lights, right? But the lights themselves won't go on unless there's actually people in the space. They have to come in and talk to the plants. When I say talk, I mean they just sort of basically blow into a CO2 sensor that's in the middle. So the idea is you have to come and sort of be there with the plants if you want them to live, to get light. Right? And then so there's this, I don't know if you call it a conversation, but there's this, sort of meeting intersection. So the lights, the people come in, they essentially trigger the lights, and those get, depending on the patterns, you hear different sounds. So it, sort of another mixer, another smart mixer. So this is sort of how it's set up. And this is how we set it up. So we had to measure the CO2 properly, these are the sensors. These are really good CO2 sensors. They're like $100 each. And they only take like a second. And And we had them in these kind of, we sort of wrap them to sort of help with the, I guess, with the airflow to get proper readings. And so these are carbon fiber, which is used for like the electrical conductive material. And these actually go inside. I have them taken out just for sort of for show, but that's the air cathode on the top, and then this goes down at the bottom. And this is Oriza sativa, Asian rice. So this was, I forget how long we had this there, but this is right around the time of the beginning of the show, and then they would grow, of course, over the next 4 to 6 weeks. I forget how long the show was. So again, it's basically the same as the MFC before, except it's plants. So yes, I call it a human-plant interface, sort of a shorthand term. But again, it's kind of this embodied flow of information, in this case, between human and plant. So I'll play the video again, but you probably understand. Just to give you a sense of sort of what the experience is like. I really wish you could hear the sound of this, because the sound is kind of crazy, because computers are fast. So it's actually switching between different clusters, different patterns that it's recognizing, like really quickly. So the lights flash and the sounds are like kind of crazy. And that's really it, right? Like it's not like a super, the idea is that it's easy to just come in and interact. Most people would blow, but I was like, talk. There's something to it. But it's just a change in CO2 levels that triggers it. So the same CO2 sensor that's up there is measuring the, inside the plant chambers. Oh, now we have sound. So let's make it a little bit of. Yeah, so we basically translated to light and sound. People seem to really get a kick out of it. Okay, so project that I'm working on right now, actually, we've exhibited it once, but we're still sort of working on it. This is called Beauty. We're calling it a machine microbial artwork. So it basically, so it involves deep learning model, what's called deep reinforcement learning. These pattern forming bacteria. And, well, that's really it, but, and I guess a robot. The deep reinforcement learning model is to control a robot. And this is yeah, so here's the robot, it's a robotic arm, so these bacteria will grow these kind of interesting patterns, and basically when there's kind of like a lack of food in their environment, so they're sort of doing like this search for nutrition. So if we prepare the plates properly, we'll get these patterns, right? Go back to so, and we're working with a few different species. Like this is Bacillus dendriformis. Actually, go back. Bacillus subtilis, Bacillus coides. So what happens is the deep learning model has a model of beauty. Beauty or aesthetics, if you like. And the play the video again. So if the bacteria, the more beautiful the bacteria look to the AI, the more food they get. Or, yeah, I mean, the more they can survive. But the idea is that the AI wants them to look more beautiful, so it's going to alter them. So we're dropping attractants or repellents onto the plate. So antibiotics, glacial acetic acid, to basically change their form, their shape. But also dropping nutrients like peptone, LB, those kinds of things, just to reshape them. So, and the sort of weird part, I guess you get to the fun part. We also have this contaminated soil that will be bioremediated if the deep learning model is happy with how beautiful they look. So here's the hook. The bacteria have to starve themselves to look beautiful for the AI. So the soil will get will get remediated. Right? So here's kind of the whole system. You know, kind of similar to the other projects, we're sort of tracking them with a camera. And we have this deep learning model, as I said. And we are kind of mapping the process to some. We actually haven't done this part yet. But that's the whole system. And then we alter their attractants and repellents. Maybe temperature in the future. We haven't done that yet either. But that's basically the system. This is a better way to understand it if you know what these things are, if you know about deep learning. So this is, we didn't build the whole model ourselves. We're using what's called the world model, which has an autoencoder, a recurrent neural network, and just a vanilla neural network for the controller. So the environment is just the images, right? And the controller learns, the autoencoder learns kind of some patterns. And the recurrent neural network is for the kind of temporal aspect, how the patterns change over time. And that will determine the actions that the controller takes. If you don't understand, you can ask me in the Q&A. It's hard to sort of do a whole summary on deep reinforcement learning, but so we're using kind of a modified version of this for our robot. Basically, they're just constructing probability distributions and taking actions based on this. With the model, it's not shown here, but that's one of the alterations. We have a model of beauty. I'm just trying to figure out how to get. So that's what I mean. How do you optimize beauty? Well, you don't. So the model of beauty is purely, as it is in the real world, contextual, culturally contextual and historically contextual. So right now, the model of beauty is just the ones that I like. We could do another one where we're thinking we could have different models depending on where we exhibited it. So it might behave differently with Korean beauty standards or American beauty standards or European, etc. So yeah, some more video of the system. And this is a project from a sort of research collective that I founded with some friends called Phylum. So if you want to learn more, just go to the website. Okay, so let me quickly, how much time do I have? Not much, right? This is the project I'm working on here. So what I proposed for the residency here is sort of a plant AI thing where I proposed to, what I said is manipulate the stress response genes, which I didn't really know much about what specifically those are. I know more now, but genetically modify the stress response genes to alter the plants, right? Their color, their shape, etc. And then train a machine learning model to recognize those and sort of tell us something about it, like, as if, again, the plants are sort of communicating with us. I've actually altered it slightly. I think I want to have the AI actually just have a continuous conversation and actually, kind of like beauty, go back and kind of modify the plant's environment. So it's sort of like this. So this is I call it Flux AI. It's just, I like to name things. But this is the AI model. Probably be like clustering. So clustering is an unsupervised form of machine learning where it just, you don't have labels, you don't have classes, so it just kind of figures out, oh, these here are similar, these over here are similar, but clusters them. So that's it. But this will be like what's called a continual machine learning model, so it'll constantly be making adjustments based on how the plant sort of reacts, right? And we'll map that to sound, just like I originally proposed. But then maybe I want to have, this is all to sort of map these clusters will be mapped to different alterations to the environment. Different water levels, temperature, light, etc. Sending it back to the plant. And what I originally proposed was this kind of, maybe like a sort of hanging garden of all of these genetically modified plants that would talk to us or via sound or text. Right? So this might get altered, but this is the basic idea of a few of these. Okay, so that's basically it. There's a lot more I could say, but what I wanted to maybe have in the Q&A, we can talk about whatever you want, but this idea of this kind of new kind of art science that, like, at least for me, it's kind of inspired by this alternative cybernetics I mentioned. And Andrew Pickering calls it, they have an ontology of unknowability. Like, we'll approach things with the idea that maybe we can't actually know everything about it, but we can interact with it. Like, I don't need to know everything about how my car works, as long as I know how to use the steering wheel and the brakes, right? I don't need to know how to actually build the car, or I don't have to know how that doorknob works, I can just grab it, right? So that kind of idea, right? And a lot of, at least the work I do, could be thought of as aesthetics of behaviors as opposed to the more formal, traditional qualities of art, where the systems, the artwork, the systems exist through the perceptual experience of the observers, of people who come to see it, right? Which again, very much aligns with past work. So it's not really about, let's say, the algorithms or software recipes, right? It's about these behavioral morphologies. And this type of work presents us with models, interesting, novel, perhaps, models of how can we engage with the non-human world and what we think about those relationships. Right? It's a different ontology, a different way of looking at the world. We typically use technology and science to dominate nature, right? To use it to just kind of have adaptive relationships. And what Pickering says is that you can kind of construct a sort of canon in contemporary art, a sort of alternative canon that uses science and technology as its substrate, and its products radically subverts the ontological premises of modern science and engineering. I would say particularly engineering in the broader sense, like not just engineering, but AI, for example, right? Which is really kind of an engineering type discipline. There's probably not enough time, but and I wanted to sort of spare you most of the theoretical sort of stuff, but this is kind of stuff I'm sort of interested in researching and writing about. To me, a lot of this stuff is this unique aesthetic paradigm is kind of grounded in these performances of agency and autonomy and adaptation and emergence, right? And that's how we get this kind of novel ontological. So I'm going to skip most of this. These are just like definitions. But maybe we can talk about this stuff, right? Like, how can we actually rethink the notion of experiment? How can we contribute to knowledge with this kind of different model, right? This Pasquian model, I'm going to call it, to bring subjectivity into science, right? And for me, I'm all about creating interfaces or windows to experience the otherness of the non-human world, or the more than human world. Right? And again, I'm not the only artist doing this, so these are just a few projects from other artists that I really like. I messed up. That's not the strong name. That's Stella Petrick. I'm sorry. Stella Petrick is this artist. She has this new project where she has an AI robot playing. Literally, the model is a play. Like they're gonna, the AI and the robot is going to play with the plants. So that's that project. This project by Andy Gracie. This is exploits traditional Asian rice cultivation. This is a really difficult project to explain really quick. But basically, he's got an AI model interacting with these plants, and it's affecting how they get managed, how they get fed. This project by Michael Sedman, he's had these photosynthetic bacteria that are interacting with another AI model using, I believe it was genetic algorithms to manage market transactions. So plants, or photosynthetic bacteria, algae, etc., they need light. So in this case, they have to buy the light from the AI model. And then in return, they give the AI model, and therefore us, oxygen. So they're sort of like, this might be another plant future, right? Where we really have to pay for oxygen. Okay, and as Javier said, there's a lot of artists involved in this field, so I'll, sort of mostly skip this, but there is sort of an active community, journals, books, all kinds of stuff. Artists are getting PhDs in this stuff, and they're involved in all fields of science and technology. So I'm really giving you like a little, sort of a little slice, right? And it's strange, right? Like, why am I here, right? What I say is that ambiguity, if we define ambiguity as kind of like emergence, that's the idea that something is always developing and in formation, so it's kind of hard to pin down, kind of hard to define. That is actually, like, get comfortable with that. That's, to me, that's that's socially, culturally relevant, and that's kind of where this work sits. The ambiguity is not by accident. So yeah, so art is an inquiry in itself, and it's autonomous. Like I said, a kind of autonomous zone of research, but it has to be collaborative, right? For kind of obvious reasons, right? And I think, yeah, we do need new ontological and epistemological models to analyze it. I gave you sort of some possibilities, but okay. So that's it. That's my work. I know it's kind of all over the place, but maybe we can have a nice discussion. ¿Hasta qué hora? ¿Está bien? ¿Hasta qué hora? Okay, está bien. So, questions, comments? You can email me, you can go to my website. Yeah.
Well, maybe I have a question. Just for clarifying something, because you tell us that the objective of your work here was to manipulate stress response genes.
Yep.
And of course, people here to know that this is a long process.
Right.
Okay?
So in practical terms, like I'm using some already genetically modified lines that people have modified. Like Javier Ramos, he has the GFP, the seeds with the GFP already that respond to different oxygen levels. Pedro has some genetically modified lines as well. So, but if I sometimes stretch myself too thin because I'm interested in so many things, but I might try another line where it would be more the stable transformation as opposed to the transient ones, right? That will just continue on even after I'm done here. I haven't quite figured out what that is yet, but yeah, there's definitely sort of practical concerns, but I kind of had to deal with the same thing with the bacteria. Right? Bacteria grow a little faster than plants, but you still have to kind of time when you're going to grow them for an exhibition, those kinds of things. So, I'm already growing plants, so I'm at least doing that. And hopefully I'll be able to start gathering a data set soon. So like Pedro's lab, they work with ABA, and so they look at the temperature, right? They use thermal cameras. So that's something I could, if I get the thermal camera, I can start taking photos of it for a data set for a machine learning. So hopefully I'll start this.
In the model of beauty that you were explaining before, so what are the parameters or what are the traits that the model takes into account to say that these colonies are beautiful or they are not beautiful?
Yeah, so the image data is just, it's an autoencoder, so this is what's great about CNNs and deep learning is the data is literally just the pixels, but with reinforcement learning, there's also like actions involved and they're connected to a time. So we're doing the images that we're collecting are time lapse. so we're training a model. We're actually, it's really slow. So our model is, our data set is small comparatively. So whenever we take a picture of the bacteria, we also do an action. Like we drop attractant or repellent somewhere on the plate. So that's part of the data set as well. It's like this action was dropped at this time at this location. So all that is taken into account.
So they need to be beautiful and smart.
And that, well, no, I mean, so that's that even doesn't even have anything to do with beauty. It's just the actions, and then the way we're using. I have to remember now exactly. The model of beauty is like this sort of thing we're adding, and I think what we're doing with it is so all we did, like I just ranked images of how beautiful they are, right? From like, it could just be like 0 to 1, you know, something like that, or 1 to 100 or whatever. And that would help the what's called the controller, the thing that actually takes the action, to compare sort of where that ranking is, right? It's like, okay, the beauty is at 50, so let me try this until I get it higher. It's really just that simple. And that just comes again from the image. It's connected to the encoder.
For your last project, when you chose Drive to do the model, was it because of something in concrete or just because some facility?
I wanted to choose something local. This was in Gwangju, Korea. Yeah, I try to contextualize it a little bit to the place. But it's just Asian rice, so rice is something we've all probably eaten it. But it happens that just outside Gwangju, they grow a lot of it. So it was easy to get. And they also had a ton of it actually at the art center itself. It's just part of, like they had a rooftop garden and stuff, so it was just kind of around. So that was the reason. Basically, it was easy and there was a lot of it. So.
And are you going to study the response of the plant only against abiotic stress or also biotic?
Well, what I proposed was,
plants infected with
I mean, when I said stress response genes, you know, that's a very non-specialist way of talking, right? So, I mean, almost anything could be considered a stress response, right? You know, any change in the environment is going to change development, right? So, but I mean, I am specifically, you know, like petals that they look at ABA, and I know that's specifically sort of like for drought tolerance and things like that. So perhaps it's better just to say environmental triggers, but primarily I am kind of looking at essentially stressing them a little bit, right? But as I said before, I'm interested in other, perhaps more longer-term research directions. Like I'm interested in callus as well, and doing weird things with them, like maybe genetically modifying them for something I don't know yet. So no, so the answer is no, not specifically just stress response. Yeah, well, I'm here if you guys just want to hang out and talk or whatever. And I'm here almost every day, so you've probably seen me around. If you're around.
Okay.
Thank you.